Good morning, everybody. The aims of the workshop today are to raise awareness about the advances in the science of error-corrected next-generation sequencing and how they can be applied to research involving mutagenesis and carcinogenesis, and hopefully inform regulatory decision-making as we go forward, not only in the UK but worldwide. And the first speaker is Dr. Jesse Salk. Jesse is a physician scientist with a keen interest in mutagenesis. He's also the co-founder of TwinStrand and helped invent and develop duplex sequencing, one of the next generation sequencing platforms that can be used to interrogate mutation in terms of mutagenesis and its contribution to carcinogenesis. Jesse actively is engaged in clinical provision of oncology treatment to patients, and so is close to the patient in terms of the impact of carcinogenesis, and therefore has a keen interest to understand the mechanisms by which cancer arises, and how those technologies can both inform clinical practice but also non-clinical safety. So with that, I'd like to welcome Jesse to the podium for his presentation. Thank you, everyone.
All right, thank you very much. So to start, we have a pretty diverse audience today. We have basic scientists, translational scientists, and toxicologists. And so my goal was to not, at the beginning, to kind of lay the landscape a little bit of some terminology and describe some kind of concepts and figures that will reference to other talks later, just to kind of get everybody on the same page. And so I won't dive into enormous numbers of details, but I think of this as a little bit of a table of contents or a synopsis to make understanding the later talks, I think, more that are really going to show data and have some more real interesting meat, more understandable. So, next slide, there we go. So I'm going to talk about starting very basic. So genetic toxicology is used to identify risks to the genome that a compound or an exposure can have that allows cells to accumulate mutations that put them at risk for malignancy, as well as inherited genetic diseases, for example. And cancer is a primary area that one is typically concerned about in genetic toxicology. Cancer is a genetic disease. It's a disease of somatic evolution, and it has to do with mutations that arise in cells, and as well as epimutations that confer a fitness advantage that let those cells grow better than their neighbors, acquire this ability to basically evolve in an adult organism. And this is a long process. Typically, when I, as an oncologist, think about cancer, I'm treating late-stage disease where there's a macroscopic tumor that's formed that's potentially spread elsewhere in the body, and this is a very nice, heroic place to intervene. But realistically, by the time you have a late-stage cancer, you're dealing with a situation which is not ideal. And so the idea of genetic toxicology and preclinical carcinogenesis evaluation is essentially to say, how can we identify chemicals or hazards or risks to the environment that can either create mutations, which put a tissue at risk for transforming into cancer, or somehow facilitate carcinogenesis, acting on mutations that either were induced or pre-exist to allow those to grow and proliferate in an abnormal way. And if we're able to identify processes that contribute to this, we could intervene or prevent them. And disproportionately throughout the kind of course of human history and medicine, even though we've made a lot of advances in treating late-stage cancer, realistically, morbidity and mortality reductions have been negligible in the grand scheme of things here compared to primary prevention and early detection. And so what many folks in this audience do and are not necessarily recognized for, because maybe you're not heroically treating somebody in the hospital is you're actually, doing something that actually from a public health perspective is extraordinarily important, and you just kind of want to call that out and recognize that. Couple of terms, carcinogenesis versus mutagenesis. I think among the lay public, probably neither of those terms are super familiar, but in the kind of more general scientific audience, I think sometimes these terms are used a bit interchangeably. And kind of from a more precise definition, a carcinogen is I'm defining this as a chemical or a process or exposure that predisposes to cancer. It accelerates the rate of growth or the incidence of cancer. A mutagen is one type of carcinogen. Things that induce mutations are generally carcinogenic at least at a certain level, but not necessarily all mutagens, or not all carcinogens are mutagens. And so if we said carcinogens is this big circle and mutagens is a subcircle, the delta, the space in between, would be non-mutagenic or non-genotoxic carcinogens. And these are two very different, from an early detection perspective or an identification of a process perspective, these are sort of two totally different things. And we're going to have some talks today that talk about detection of mutagenesis, which is a risk factor for carcinogenesis. We'll also have several talks that describe kind of more broadly detecting carcinogenic processes in a way that are maybe mechanism agnostic. And so just want to really think about that. Mutagens act, again, at an early stage, and the increase in selectable diversity in the tissue can increase the probability of developing cancer, but also, even in the absence of adding more mutations, compounds like estrogen or androgen mimetics, PPAR agonists, a variety of things that kind of create proliferation, even though they're not directly mutagenic, can contribute to this process. And I want to just kind of help people to think about those as two distinct things. They can be evaluated with similar technologies, but in different ways. Generally speaking, I think most people in this audience have some sense of next generation DNA sequencing because you're here, but just to establish a baseline, there are a lot of different tools. There's a term next generation sequencing. That's a bit of an anachronistic term because it's been around for a while. And then how do you go forward if you're already at next generation sequencing? People didn't think far ahead. So I'm just calling it kind of contemporary or modern DNA sequencing. Generally speaking, and this is just kind of a one major kind of workflow. This is not exclusive, but generally one takes DNA and you chemically extract it from a tissue, and you're just using chemistry to isolate nucleic acids. We typically fragment it into little pieces. A common approach is then to ligate on what are called adapters, which are just sort of universal primer sequences that let you kind of work with this heterogeneous population of molecules because you have a common sequence on them that can stick to the sequencer. Commonly, we then use that to do PCR amplification of this population. Commonly, we enrich for a portion of the genome we're interested in, but one doesn't have to. One can work with the entire genome. We'll have talks from Dr. Martin Carina describing something along that lines. We'll have a talk later related to CRISPR mutagenesis, where amplification is not done as this process, but this is just, again, kind of a general example. Then one takes these molecules that have this universal sequence and it puts them on a sequencer, and this is kind of most of these sequencers are effectively like a microscope slide that have a lot of DNA molecules in the surface that are kind of compatible with the universal sequences on these DNA molecules. And then there's a kind of what's called a colony amplification, which is kind of like, think about streaking out a plate of bacteria. They're invisible, and then you let them grow, and where one bacteria landed, it forms a colony. This is sort of a biochemical colony. And then these colonies, which are initially invisible, are sequenced by virtue of sort of flowing different reagents across. And when a certain nucleotide is appropriate to be incorporated at a given sequence, it incorporates a fluorophore, a certain color. And so you'll have lots of little spots. Sometimes they're randomly distributed. Sometimes they're in a patterned way, depending on the sequencer. but you'll just have these different colors. And if a given spot goes red, green, blue, blue, red, that means AT, CCG, or whatnot. And then the computer on board the sequencer converts those fluorescent kind of spots into sequence reads, which come out in as strings of data, AGT, CCT, and there's a sort of certain confidence that's associated with each one of those bases, and then to figure out where in the genome they belong, we typically do something called DNA alignment, which is you simply take each one of these millions upon millions of sequence reads, and a very sophisticated computer algorithm just goes through the genome and says, this has a best match in this particular place, and that creates an alignment or a pileup. And then from that, we'll say, well, compared to the reference sequence we're looking at, is this sequence exactly the same, or is there a slight difference? And that's basically what we're doing. And you'll hear from Clint Valentine later talking about informatics in more detail and others, but just want to give that kind of orientation. We're mostly going to talk about at the level, forgetting about this stuff, at the level of the data, what do we do with it? Conventional DNA sequencing is very, very powerful. The most common sequencer is manufactured by a company called Illumina. They have, 90% market share. And so most of the data you'll see today is coming from Illumina sequencers, although there are new sequencers coming out that are some are similar and some work in different ways. All these sequencers operate on kind of similar principles. And sequencing has gotten much less expensive over time. We can sequence megabases. So megabase is a million base pairs, like a megabyte, million bytes, megabase, million bases. And we can generate gigabases or even terabases, trillions of bases of sequencing off of a standard sequencer now. And this lets us sequence the whole genome. The first human genome took hundreds of millions of dollars to sequence for one person. Now we can whip out, Sanger has, the capacity to generate, 1,000 whole genomes a day, and they don't even work very late. Just very, very standard European hours, which is, I hear a point of contention. So very impressive in terms of the scale, and that lets us look at all sorts of things we weren't able to see before. The genetic differences between one person and a population that puts them at risk for something, and we can sequence a tumor and differentiate from a normal in a given person and say, what are the genes or mutations that are responsible for driving that cancer? And it works great for that. And so we can learn an enormous amount, and you'll see a lot of data later from folks at King's College and Sanger and others looking at things called trinucleotide mutation signatures, where we can take these patterns of mutations and infer all sorts of things about what happened with the tumor. And it lets us kind of do this archaeological process. We look at the tumor and we can kind of infer what happened to it. Mutagenic carcinogens can leave very specific patterns. Non-mutagenic carcinogens maybe don't leave the same kind of mutagenic patterns, but they can maybe select certain mutant genes in a different way. So we can tell a lot about what happened to cause a tumor, archaeologically speaking, looking backward. But one of the challenges to that is that we're sort of looking backwards. This is a type of data you'll see a lot from other folks. Again, this is an example of what's called a trinucleotide mutation signature, mutational fingerprint. And think of this almost like a barcode. You can use some fancy branches of math to sort of look at this pattern and say, is it similar to another pattern in a database that's caused by something we kind of know? And it's, think of it a fingerprint or facial recognition. So very, very powerful tools that have been developed. And really much of the leading science has been actually out of the UK and some of the folks you'll hear from today. But this really works when a cancer is already formed. And so with a standard sequencer, one has to have kind of the majority or a substantial portion of the cells in a sample kind of carrying the same mutation for it to be identified. So a tumor is a clonal population. It started from, presumably a single cell, and all the derivative cells kind of are derived from that same cell. There will be other mutations that accumulate along the way that are subclonal, but generally speaking, think of a tumor, you show these ugly cells with this, purple color as a clonal mutation, and the reason that mutation is in all the cells is because they came from the same place. If one is looking in tissues that are not clonal, Well, and that's also a hard thing. Every tissue in our body is clonal. We all came from a single fertilized zygote. But let's just say we've grown into an adult organism, and then one cell gains the ability to proliferate abnormally. That starts with a single mutation, and if that hasn't expanded to represent a majority or a substantial portion of the sample being sampled, the sequencing technology can't pick that up. So we can identify these mutational processes from clonal tumor populations. We can identify them from artificially created clonal populations. You take a single cell, you grow it in culture, and then you sequence that. But this is sort of either an artificial system or kind of working backwards. And if we, and by we I mean many of you as toxicologists, are wanting to say what is the risk of this thing, you want to look from the other side. Cancer risk assessment is about the future, not the past. And in terms of genetic toxicology, we have a lot of historical assays, things like the Ames assay, transgenic rodent assays, which you'll hear about from Bob Young and others today, where these are tools for detecting very, very low frequency mutations that are caused by mutagenic chemicals. And historically, we've not been able to, we don't have technologies that are accurate enough to sort of just look through millions of cells and find those mutations. So all of these historical systems are based on selection. Some mutation that allows a cell to grow better. An Ames assay, a mutation that happens in a large population that overcomes the oxytrophic limitations and survive and reproduce in the absence of the provided nutrient. And those assays sort of don't give us this richness of information of these mutational signatures and others. And it would be just lovely if we could use these next generation sequencing tools to look at that level of data that we can see in tumors, but have the ability to see it at these very early time points. And that's really what this kind of lecture, this discussion today is going to be about, is using these new technologies in a special way to get that information and get it in a faster, simpler way using modified versions of these sequencing technologies. Standard DNA sequencing typically is, think of it as looking at clonal mutations or relatively clonal mutations. And I'm just showing you here an example. This is a 400 base pair region of the human genome. Each tick along this axis is a nucleotide position, and the height of each bar corresponds to what we call the variant allele fraction. And that's a term you'll hear today. Which means the percentage of molecules in this population being sequenced that carry a specific variant or mutation. And in the situation where you're looking at an inherited variant, like a heterozygous or homozygous variant, you'll see 50% or 100% of your molecules carrying that. And that's very easy to detect with standard tools, and there is a little bit of background noise, which is this red here on the bottom, but it's sort of irrelevant at the scale of looking for clonal mutations. And this is true in tumors, and this is true in cloned single cells. But if you zoom in the y-axis and put a maximum of 1% rather than 100%, you can see that this noise becomes relevant at this scale. And if you have a true mutation that's present in 0.1% of your sample, for example, you wouldn't know if that's real or that's not. The background mutant frequency, and we know this from Ames assay, we know this from HPRT, in sort of mammalian cells or bacterial cells, is very low, on the order of 1 in 100,000 at the very, very highest, really, usually 1 in a million, 1 in 10 million. And so this is orders of magnitude too high a background to be able to detect that type of mutation. And so this concept of error-corrected next-generation sequencing came about, which is a way, a kind of a group of ways of saying, how do we improve the accuracy of these machines? And unfortunately, these machines have sort of asymptotically approached the level of accuracy that's possible to achieve based on the optics and the chemistry and whatnot. And so it's, now we're looking at 1 in 1,000 error rates, for example, but that's still really far away from 1 in 10 million. And so this class of technologies is not so much about modifying the sequencers themselves. It's about using some process whereby you're kind of duplicating the molecules you're working with and labeling them in some way that you can tell which of these duplicate sequence reads came from an original starting molecule, and then say, well, any variation among those sequencing reads must be an error because they all were sort of clonally derived, and you use one or more techniques to kind of label that original starting molecule before you do the manipulation to kind of subtract out errors. Duplex sequencing is the one that twin strand is based around. It's a very, very high accuracy version that relies on comparing the two original strands of DNA molecules. One of the first published methods, prior to that, was something out of Bert Vogelstein's lab called SafeSeq, which makes copies and creates consensuses around sort of targeted single strands. You'll hear from Dr. Martin Carina, NanoSeq, which is a advanced flavor of duplex sequencing using some additional elements. There's and primarily focused on very low depth sequencing, but covering a large swath of the genome. There are nanopore sequencing techniques. There are single molecule consensus making through a platform called PacBio. And there's a lot of these, and I'm not going to get into them in detail, but I just wanted to give a sense of what those are. Duplex sequencing is a technology where we are labeling large numbers of original double-stranded molecules in such a way that we can differentiate the sequence reads from those of other molecules. We can relate the sequence reads together, those that came from the same original, the two strands of original starting molecule, and also tell them apart. And so typically we put on these special molecular labels, we make copies, we'll enrich for a portion of the genome we want often, and say everything that came from the original top strand, the original bottom strand, should have the same sequence, and any variation between those must be an error. And we can create a double-strand consensus sequence and say the only true mutation here, A opposite of T, is present in most or all the copies of both the original top strand and bottom strand. Its complementary changes at the same position, and that lets the error rate go way, way, way down. So this is an example, standard sequencing and duplex sequencing. This is y-axis of 1%, and you can see that there's a lot of background noise that goes away. There's only one true mutation. In this particular case is an example of a clinical example of a patient with a pre-existing drug-resistant mutation on a particular targeted cancer therapy who ended up relapsing several months later, and this low-frequency mutation underwent selection, and had I known that as an oncologist, I would have, in this case, theoretically been able to switch to a later generation tyrosine kinase inhibitor and potentially prevented that. So that's just an anecdote, but you can see noise reduction, very important for some applications. Some basic nomenclature, we're in time, just to also orient people. Coming back to this clonal mutation, I'm defining as a mutation that's present in all or most cells of a sample. And if I sampled your entire body, I took you and put you in a blender and sampled some DNA out. I'm only going to see the germline mutations, things that are present in that same fertilized zygote. In a tumor, if I took the entire tumor, and I would only see mutations that were in that single sort of founding cell. But if I took small pieces of that tumor, there are probably subclones within that. As that tumor expanded, new mutations arose. So that's kind of what we're sort of saying a clonal mutation is, and standard sequencing is very good for that. A random mutation, I'm defining as a mutation that's present in one or a very, very small number of cells. Say, one molecule of benzoate pyrene from breathing smoke on the road got in and created an adduct and caused a mutation in one cell. And if I did standard DNA sequencing on a population of cells, I would never identify that because that's below the background mutant frequency measured by the sequencer. And a subclonal mutation is something that's kind of in between those two. It's something that we say is in a proportion of cells, but not all of them. And this is the kind of the most complicated idea because a subclonal mutation can get in a tissue through a few different ways. It could have been something that's a very mutagenic part of the genome that recurrently independently mutates in many single cells over and over and over. It could have been in a single cell and a mutation in, say, RAS, which allowed that cell to grow and clonally expand, and therefore it's a driver of sort of a carcinogenic process. Or it could be a random mutation that happened to be in the cell that had a driver mutation elsewhere and get carried along as a passenger. So interpreting what this means is kind of complicated, and you'll hear different talks describing each one of these differently, but just want people to kind of get that sense. There's obviously a bit of a continuum between these, but if we're going to bin them logically, that's, I think, relevant. to think about. And I'm going to show maybe three or four more slides, just showing you some of the types of data you're going to see from other talks. One can measure a few different things. If one is talking about mutagenesis and using an error-corrected sequencing process, mutant frequency is the kind of simplest thing you're going to see the most data about. And mutant frequency, we're defining as number of mutant nucleotides divided by the total number of nucleotides sequenced. And with these error-corrected sequencing approaches, one nucleotide of data corresponds to one original nucleotide of an original molecule, because we sort of remove these kind of artificial duplicates in the process. And so this is data, and Bob Young will probably talk about this in a bit, from a paper we did not too long ago, where we're looking at an animal sample that was treated with chemical mutagen urethane in several different tissues. Each bar is a different animal, a biological replicate. And you can see, for example, in lung, the VC, vehicle control, has a low mutant frequency, and it goes up with treatment with urethane. The way we're calculating these numbers, in this case, for the very first sample, I'm just showing you what the calculation was. We sequenced 189,664,560 nucleotides, error-corrected nucleotides of data, and we found 16 mutations. Numerator divided by denominator equals frequency. And this error bar is a statistical confidence that can be sort of inferred based on binomial counting statistics. So if we had let's say 32 mutations divided by twice as many nucleotides, that error bar would have come down. This is counting statistics sampling. These error bars you'll see sometimes are not about variation among replicates. It's sort of counting statistic error bars. When you can plot mutant frequencies, and you can see changes over time with different doses or different times or different tissues, and these are just some of the kind of ways you'll see people present data. We can look at what's called a mutant spectrum. And a spectrum sounds complicated, but it's really just a frequency of a subset of mutations. This type of plot we call a simple spectrum plot. And we're dividing up the different mutations. So all the mutations found in a given sample, each a T to G versus a T to C mutation have different colors. And then in this case, we're measuring the proportion of each. And we're saying, well, in this sample, 1.18% are these blue ones, and whatever percentage are those. And we can then take that data and put it into computer algorithms that can look at these patterns and say, these samples look different than these samples based on this approach of unsupervised hierarchical clustering. But fundamentally, we're just looking at a frequency or proportion of a subset of mutations. The colors in that got funny. Well, you'll hear a lot more about this later, but trinucleotide mutation signatures are the same concept. It's just divided even more finely. So in this case, rather than looking at, say, 6 different subtypes of mutations, say C to A versus C to T, we're also looking at the adjacent nucleotides, because the adjacent nucleotides have either more electronegative or positive, or somehow change the kind of chemical structure and predisposed to a given sequence context being mutated more frequently than other. This is urethane. Urethane has a very characteristic mutation at the motif NTG, ATG, CTG, TTG, or GTG. And you can see that you have this particular pattern, and then other chemicals, we don't see that. And so subdividing helps us identify these patterns. But again, each one of these is just a frequency or a proportion. So it all comes back to the same concept. And then the very last concept is that carcinogenesis. So carcinogenesis is, remember I talked about how mutagenesis is different than carcinogenesis. Carcinogenesis is a broader process or picture. Mutagenesis is a subset of it. It turns out that we don't have just random mutations or just clonal mutations. You can have a situation, and this is probably true in many, if not all, cancers, where there's some early cancer clonal evolution process that doesn't lead to ugly cancer cells, but it actually, at the level of the cells, there has been some clonal expansion that's gone on, but not enough, and not enough mutations have kind of accrued to create an actual tumor. And in theory, if we could identify these expansions, that would give some inference about carcinogenic processes going on, whether it's due to mutagenesis or whether it's due to a non-mutagenic carcinogen. And so these are kind of, a term in medicine or pathology we historically use called a field defect, an area of a tissue that's been sort of predisposed and sort of partly transformed into along that cancer process. And so you might say, well, how can we use a DNA sequencing tool that looks for low-frequency mutations to identify non-mutagenic carcinogens? And the answer is we can, but we do it differently. We're not looking for induction of mutations. We're looking for the ability of cells to expand by virtue of identifying subclonal mutations, something that is carried into a proportion of cells in a gene that is historically been known or recognized as being associated with cancer. So Bob, and we'll talk about this, and Patricia Escobar later today from Merck, but the idea that we can treat animals with a carcinogen, and rather than looking for random mutagenic processes, we can look in genes that are sort of historically known to be associated with cancer and find certain hotspots that are under positive selection and look for clonal expansion of mutations in that particular tissue. And by virtue of seeing something as not a random single mutation, but as a subclone, a proportion of molecules, we can infer that a clonal expansion event had gone on, and that would be relevant to inferring carcinogenesis. And then we can look at both of these together. We can look at mutagenic processes and carcinogenic processes in the same tissue, and you'll hear a bit about more about this later as well. So that's the overview. I'm really excited to hear all the other speakers coming on.